Tonight's speaker has fundamentally shaped our scientific understanding of human memory formation. Lynn Nadell's research focuses on the role of the hippocampus in memory and cognitive mapping, with a particular emphasis on episodic memory and context. Lynn has advanced the multiple trace theory that the hippocampus is always involved in storage and retrieval of episodic memory, but that semantic memory can be established in the neocortex. He also investigates aspects of hippocampal development, the impact of stress on hippocampal function, the nature of spatial cognition, and the contribution of hippocampal dysfunction to mental retardation in Down syndrome. And he has begun several projects involving the interaction between learning, sleep, and memory consolidation. Lynn and John O'Keefe co-authored the influential 1978 book, The Hippocampus as a Cognitive Map, which defended the theory that the hippocampus learns and stores cognitive maps of portions of space. Lynn and John together won the 2006 Grawemeyer Award for their work in identifying the brain's mapping system. Lynn received his PhD from McGill University in 1967 and held research positions in Europe, Canada, and the US before he joined the faculty of the University of Arizona in 1985, where he currently holds the position of Regents Professor of Psychology and serves as Director of the Cognitive and Neural Systems Program. He has authored or co-authored more than 175 papers and 24 books. He is a Fellow of APS and the American Association for the Advancement of Science. Lynn has titled his keynote address, Making and Remaking Memory, Past, Present, and Future. Please join me in welcoming Lynn Nadell.
Well, first of all, I want to thank Suparna and APS for inviting me to give this, this prestigious talk. I hope you can, I need to plug this thing in. And I also want to thank those of you who are sticking around to hear it. I can't see any of you, basically, so you can feel free to leave without worrying if I see you leave. I understand that I'm standing between you and dinner. Those of you who know me well know that I'm also standing between me and dinner. So I'll try to make this as concise as I possibly can. Are we up? Great. I think I can wander around. I have one of these fancy things that allows me to move, I hope. Once I get started, it'll be quicker. All right. So, is this going to work? Yeah. I could tell the story about memory. I could go back to not just William James, but to Aristotle. In some of the versions of this talk, I've done that. That's not a good idea tonight. I'm going to jump to the sort of what I take to be the beginning of the modern era, you might say. It's probably not really the beginning of it, but it's a signal moment for many of us who are interested in the sort of brain mechanisms of memory, which I was, with HM. Now, most of you know this, but I'm going to tell you a few little details of the history of the HM story that you might be unaware of. So this is HM. We now know his name. This is the surgeon who did the operation to control his uncontrollable epilepsy. The surgery actually did kind of work, but it rendered HM profoundly amnesic, as you all know. Scoville, the surgeon, that's the surgeon and the presumed lesion that was made, didn't really know how to deal with this and contacted one of his friends, Wilder Penfield, at the MNI, who had recently published something about similar patients. Penfield, and asked him, do you want to study this patient? And Penfield passed this request on to his colleague, Donald Hebb, who, okay, that's interesting, and Hebb passed this request on to his student, Brenda Milner. So this is a picture of Brenda Milner, kind of roughly speaking, when she was a student. Here's a picture of Brenda more recently, not so recent. Here's a picture from 3 days ago of Brenda Milner receiving the sort of honor of award from the whole government of Quebec on the occasion of her 100th birthday. So Brenda's really a remarkable individual. You can applaud for Brenda. Brenda then passed it on to Sue Corkin, who spent her entire life studying HM. Sue unfortunately passed away a few years ago, but not before she wrote this fabulous book about HM that I would recommend to anybody who wants to understand what it was like to work with a patient like HM to make a career in some ways out of that. Now, small historical note for those of you who think about how small the world is. Sue Corkin married my college roommate in 1961. That's how I first heard about HM, prior to even becoming a graduate student in psychology. All right, so in understanding HM, you have to understand the way HM's amnesic problems were interpreted within the then existing thoughts about how memory worked. This is the way people in the 50s and 60s were thinking about memory. I think Rich Schifrin's in the room. He'll recognize this only too well. The notion was that events happen and they trigger some sort of an immediate memory, echoic memory, iconic memory. Some of those things get attended to. They end up in short-term memory. A lot of that gets forgotten. Some of it gets consolidated into long-term memory. And by this view, different memory systems differ primarily in terms of duration. Sort of how long are they processing and storing information for varying periods of time. And you can fill in some times there that, roughly speaking, are correct. Of course, the short-term memory buffer can be rehearsed. So this was the framework within which HM was interpreted. And in fact, in the original paper, in the Atkinson and Shiffrin model, there was references to HM. They were thinking about HM when they were thinking about how to interpret what they were talking about. So this early work with HM was taken as support for this particular view of memory, because it seemed in the initial work with HM that he had normal immediate memory. He had intact short-term memory, because if you didn't distract him, he could hold on to a few bits of information and give them back to you. He also seemed to have relatively intact long-term memory for things that happened prior to the surgery, or at least that was what people thought at the time. Turns out that's not quite true, but that was what was thought at the time. So given that picture of what HM looked like, the problem appeared to be a problem of getting, I think I have a pointer here, but it doesn't show up on that screen. Okay. The problem appeared to be getting information from short-term memory into long-term memory. In other words, it was kind of a problem of consolidation. So since HM surgery largely excised the hippocampus, this part of the brain was assigned a role in memory consolidation. Immediately, animal models were attempted. Again, in Montreal, that's Brenda Milner as well, along with Jack Orbach, who was, I think, maybe Hebb's last graduate student or postdoc, and Theodore Rasmussen, who was a neurosurgeon in Montreal. They created a monkey model trying to replicate exactly the same lesion that HM had been subjected to, and lo and behold, those monkeys showed no evidence of an amnesic deficit. There was a complete failure to replicate in this monkey model what had been observed in the human case. And the same thing happened in people working with rats. They made lesions in the hippocampus, didn't appear to affect learning and memory in any general way that mapped onto what was observed in HM. So those are some of the original work. And the question is, why not? Well, I think, and I hope to show over the course of this talk, that they were working with some sort of an inappropriate notion of how the brain organizes memory, but I may or may not be able to make that case. Well, because of this, and I was a graduate student exactly at this period of time, there was this split between thinking about what the hippocampus does in humans and what it apparently does in animals. This split led to chaos in the hippocampus world. I mean, and I really mean that seriously. There was sort of this chaotic sense of we have no idea what this structure is doing. You get very different results in these two different species. And just to prove the point, I'm going to show you a picture of a meeting of the Hippocampus Society from roughly that period where people were debating what was going on. It was a mess. Okay, that didn't really happen. Anyway, roughly speaking, 1964, there's the rat hippocampus, there's the human hippocampus, and people were saying things like, maybe they do something radically different in these different species. Maybe this structure of the hippocampus has come to do something very different than what it was doing in the monkey and the rat. Well, this guy would differ. This is a young Charles Darwin. It just doesn't make any sense that a structure that has evolved over 350 million years is suddenly, between non-human primates and primates, is going to have a totally different function. That just doesn't add up. So the field stayed in this kind of chaotic zone until a critical discovery was made by John O'Keefe, when he discovered place cells in 1971 with his graduate student, Jonathan Dostrowski. So this discovery, and I was a graduate student with John. He was also at McGill, and we were good friends, and I ended up in London sort of in a position just down the hall from John. We were happy to talk to each other. We weren't working together. He came down the hall and showed me the initial data, and this is kind of like winning the scientific lottery, essentially. You work down the hall from somebody who makes a Nobel Prize-winning discovery, and they ask you to join in. That doesn't always happen. So John and I then began to collaborate on trying to make sense of this discovery, and we spent 6 years writing this book. Now, most people think about this book, to the extent to which anyone thinks about it, most people think about this book as a book about space. But that's a mistake. That was actually never our intention. That was only about space. Here's a quote, not from buried deep in the book. This is on page 1. In fact, I think this is in the first paragraph. And it says, I believe you can read quite clearly, that we're talking about a memory system. It's a memory system of a very certain kind, but it is a memory system. So even though most people still think about this as a spatial theory, it's really a memory theory as well. So I'm going to talk for the rest of the talk, and I have about 3 hours, right? I'm going to talk about the implications of cognitive map theory for the way we think about memory, and I will try to be, as concise as I can. So this theory, this approach to thinking about memory, has implications, makes certain statements about how memories are organized in the brain. But it also says something about what is memory for. Why do we have memory? What purpose does it serve? So early on, this comes from an even earlier paper that John and I wrote in '74, we talked about memory and this cognitive map as providing predictions, that the point of memory was to make predictions about what if I do this, then this will follow. If I go around the corner, I'll see this, and so on. That memory is always about making predictions. Okay, this, by the way, is just Tolman redux, for those of you who are familiar with Tolman. So how is memory organized in the brain? The first clue, and this is again something that we stressed in the book, there is not one memory system, there are multiple memory systems that do different things. Here are some quotes from various things that we wrote back then that make this point, that not all memory is stored in the hippocampus. Memory systems that are concerned with other things like the value of objects, with motor habits, with procedures. This is kind of new now. Sorry, this is not new now. Everyone understands there are multiple memory systems, but back in the 70s, that was not the way people were talking about it. And the point we were trying to make here is that these multiple memory systems differ by virtue of the kind of information they process and store, not simply how long they process and store it for. That's a key distinction. So again, here's another early quote, no such thing as the memory area. There are different memory areas, each of them responsible for a different form of information storage. And we assigned a particular task in this zone of memory to the hippocampus. It constructed and stored these cognitive maps. It wasn't important for learning that this room would get shocked, sorry, that this object would shock you, or it wasn't important for learning what apples taste like, but it was important for constructing and storing cognitive maps. So this theory then suggests that specific kinds of information are processed and somewhat represented in specific parts of the brain. This is a little bit simplified now because we now tend to think more in terms of networks than sort of little blobs, but nonetheless there are hotspots, so to speak. Our focus was on the hippocampus. Again, there's the rat and the human hippocampus. The notion is that the hippocampus represents information about space and episodes. And because of that, because it represents and processes that kind of information, it supports behavior that is dependent upon contexts and places. And that without the hippocampus, you would not be able to behave very effectively with respect to those particular that particular kind of information. So here's those of you, again, who know about Tolman, here's an example of what we meant by this. So these memory systems differ in terms of the behavioral strategies they can enable. So here's a kind of a plus maze or a T maze. Typically, rats are trained to start at the bottom, well, the bottom of the slide, and to make a right turn to get some food. Now, they could actually learn this either by learning to make a right turn, or they could learn this task by learning to go to that place in the world. You don't know which if you stop the experiment there. However, what Tolman and his students intelligently did was start the animal from the other side. Now, if the animal learned to make a right turn, it's going to do what I just showed you there. That's called a response strategy. If the animal uses a place strategy, it's going to do this. And what we were suggesting was that the hippocampus is crucial for that kind of strategy. All right? So we did an experiment. It's usually a good idea. We did an experiment to sort of assess this notion that the hippocampus is crucial for just a particular way of learning in the world, and that for other kinds of learning you don't need it. So here's an apparatus that we designed. It's kind of an annulus that's an open to the world. The animal can see where it's going. You see those little white dots in that annulus? Those are water wells into which we could pump water. One of them, the green one, it's not marked green, though, just for your sake. If the animal goes to that one and sniffs at it for a few seconds, we then would pump water up, and the animal would get rewarded. So it had to sort of find the reward by knowing where to go in space. There was nothing that identified that particular location other than its position in the broader world, and the animal could see things out in the extra maze environment. So what happens when you train or try to train animals with damage in the hippocampus on this task? Here are the results. Basically, animals, we did fornix lesions, were incapable of running this task. They simply couldn't do it. It's a relatively simple task for a rat that has a whole brain. For a rat with damage to the hippocampus, couldn't do it. But, again, our position was that it's only this kind of learning that you need the hippocampus for. So then we did the following. We then put a light and shone that light on that hole. So now we have something else identifying where the reward is going to be. Now, immediately, this required no further training. Immediately, the rats with fornix lesions were capable of solving this task. No problem whatsoever. We then did the control, which was we now move the light around. We give the animals a choice. And what you find is that the control animals all went back to the original place where they'd been rewarded. The animals with fornix lesions virtually all went to where the light was. So these animals were performing this task using different behavioral strategies. And the hippocampus was critical only for one of those, the place strategy. Okay. In addition to sort of supporting different strategies, these memory systems also differ in terms of developmental trajectory. This is a slide that I took from Christina Alberini, I think, but you can see from this slide that some aspects of learning are delayed in sort of maturational terms. And the point that this makes is that hippocampus-dependent learning is not available immediately at birth in most species, in so-called altricial species. In some precocial species, like the ones that are running around the world as soon as they're born, they have a functioning hippocampus right away. But animals like us and most mammals, their hippocampus shows a delayed maturational profile. Well, that's interesting in the context of the selective role that we assign to the hippocampus. So for what we know is that the human hippocampus function seems to emerge only after 18 to 24 months, and adult function perhaps as late as 10 to 12 years. They're still sort of getting better and better at the things the hippocampus does. I'm not going to go into detail on any of these things, but I just want to make a few points. What this means is that for the first 18 to 24 months of life, humans don't have this, don't have the things that the hippocampus can give you. So that should lead to, and in fact it does, lead to an inability to form episode memories. Infantile amnesia. Freud first talked about infantile amnesia. He talked about the various ways it could be caused. He included sort of biological immaturity as one of the things that could be leading to infantile amnesia. He didn't focus on that. But it looks like that's part of the story, a very important part of the kind of hard infantile amnesia where virtually none of us can retrieve episodes from the first 2 years of our life. And if we do retrieve them, it's because we were told about them. There's also a delayed onset of the core functions of the hippocampus, this exploration and place learning. Exploration is what an animal does, what we do to build our cognitive maps, and it's a core function of this system. Those things are also delayed in onset in most species that have been studied. In addition, because the hippocampus is still in this kind of active developmental stage after birth, it's sort of more susceptible to environmental perturbations. So early stress, early nutritional challenges, all of those things can actually influence the development of the hippocampus in negative ways, sometimes in positive ways, but typically in negative ways. And indeed, chromosomal disorders like Down syndrome, where the burden of this extra chromosome is there right from early from conception, but from birth, it appears that that kind of a syndrome preferentially impairs late developing structures like the hippocampus. So we've been able to show that the individuals, toddlers with Down syndrome, have sort of particular deficits related to hippocampal dysfunction in a way that might be amenable to treatment down the road, and we hope that to be the case. And mouse models have confirmed a lot of that. All right, so here's a summary of the first, I hope, third of my talk. The brain organizes memory in a distributed fashion. There are multiple memory systems, each of them representing a different kind of information or knowledge. These different memory systems have distinct properties. They're grounded in physiology. I left the slide out, which sort of allocates rapid learning to the hippocampus because of the fact that LTP, this long-term plasticity mechanism that was discovered in the 1970s, was discovered in the hippocampus, leading people to think that it was the structure critical for sort of one-trial rapid learning. They differ in terms of developmental trajectory, and they differ in their response to various neuromodulators. I'm not going to say anything about that here. The other thing that I want you to note is that there is this deep connection between memory and space. I mean, we're talking about a system that is doing something fundamental both in terms of memory, but also in terms of space. And finally, and this is what I'm going to turn to now, memory consolidation. This was what the hippocampus was initially thought to be important for, and it was thought to make a memory permanent. It's not quite what we thought it was. So I'm going to segue now to the issue of memory consolidation, sort of how are long-term memories actually made. Well, again, we go back to the notion something happens, there's an immediate memory, you attend to some of it, short-term memory, consolidation, long-term memory, and so on. What's the role of the hippocampus here? The idea, okay, this just repeats what I showed you before. The idea was that the hippocampus served as a kind of an intermediate-term memory system that held on to the information or retained the capacity to retrieve the information during this consolidation period when long-term memory was being built up in cortical circuits outside the hippocampus. That was the idea. And that as time passed, as consolidation time passed, this memory outside the hippocampus would become strong enough that it could self-sustain and self-retrieve, and you no longer would need the hippocampus. And you would recycle those neurons back into the pool to be used again for some future experience. That was the general idea. And this led to the so-called standard model of consolidation. I was, along with Larry Squire and Neil Cohen, involved in, I think, one of the earlier sort of formulations of this. And the notion was that the hippocampus was important for explicit memory. Now people were talking about explicit and implicit memory. Notice we're now in the 1980s, so the idea of multiple memory systems has now entered the field. So the hippocampus was important for explicit memory, but only initially. That over time, memory expression became independent of the hippocampus, either because the memory was in the hippocampus and it got moved somewhere else, which is a completely bogus idea biologically, by the way, or by transfer of control over memory expression, that the memory was always in the cortex, but it just couldn't be retrieved efficiently without the help of the hippocampus until it sort of self-organized in a way that it could be retrieved. Well, and the idea was that in some way reactivation of the memory, possibly during offline processing or during sleep, led to this consolidation process. That was, roughly speaking, the notion. That idea about sleep came later in the McClelland, who Jonathan mentioned. Jay McClelland, Bruce McNaughton, and Randy O'Reilly published their quite influential paper on two different kinds of learning systems. The problem with this approach was there were some significant issues with the standard model of consolidation. I'm just going to mention two of them. One of them was the length of retrograde amnesia. How long prior to the injury were memories lost for? In other words, how long was the consolidation period, so to speak? How long were memories still fragile such that when you injure the hippocampus, they could no longer kind of finish their consolidation? The data that were emerging in the early 90s were indicating that this period of time was way longer than it should be. It wasn't a day, it wasn't a week, it wasn't a month, it wasn't a year, it was getting to be 10 years, 15 years, 20 years of these very long retrograde amnesias that just didn't fit within any sensible model of consolidation as a system that would recycle neurons back into the usable pool. If it takes 20 years to do that, and you're talking about a system that evolved when lifespan was on average 35 years, it's probably not a temporary system, right? So that was one problem, and Morris Moscovich and I sort of puzzled over that. We also were puzzled by the fact that it appeared, contrary to the standard model, that episodic and semantic memories suffered rather different fates in amnesia. that sort of semantic memories could be saved, but episodic memories couldn't in some way. So both of those led Morris and I to propose the multiple trace theory. It was built largely on the cognitive map theory, but also incorporated some elements of things that Morris Moskovich had been working on in the early 90s, sort of automatic processing in the hippocampus and so on. So the multiple trace theory made a few critical assumptions or assertions. First of all, that the hippocampus plays a role in storing and retrieving episode or context-dependent memories even when they are remote, but it plays only a temporary role in storing and retrieving semantic memories, so pulling those two kinds of memories apart. The hippocampus, as I've already indicated in this view, represents spatial or contextual relations in a way that's not reproducible elsewhere in the brain. So the idea here is that you can't just take the hippocampus out and imagine some other part of the brain doing what it did completely. It may partially substitute for it. It may give you a workaround, but it is not going to do exactly the same thing the hippocampus did because it's not wired up the way the hippocampus is wired up. It doesn't have the connections the hippocampus has. It's a different beast. One critical added assumption. When you retrieve an episode memory, this initiates new encoding that can alter and expand that trace. This is the critical assumption that came to be the most important part of the theory. All right, the theory made three core predictions. They're there. Essentially, they're related to what I just said. The hippocampus represents certain things. It does this no matter how old those memories are. And every time you retrieve one of these memories, this results in some sort of activity that can sort of alter how that memory is represented. Memories, in other words, are malleable in ways that people hadn't previously paid much attention to, with the possible exception of Beth Loftus and people working sort of a bit at an angle to the things that those of us were doing. So here's one experiment. I'm going to run through quickly a few experiments that address these 3 points. In this experiment, we brought people in for an interview. We asked them to recall spatial and non-spatial details for a bunch of personal events. Some of them were recent, some of them were remote. No, I don't think that's part of this experiment. Drop that. They were all pretty recent. And this interview allowed us to construct 4 different kinds of questions, which the people then got in the scanner as true-false questions. We wanted to make them think about things that were both spatial and episodic, or non-spatial and episodic, and so on. And then to ask the question, which of these conditions is the hippocampus most interested in, most activated by? Well, the results were, as you could guess, I wouldn't be telling you about it if this wasn't the case, that there's a main effect for space. The hippocampus is the red bit, and the yellow bit is parahippocampal gyrus. The hippocampus and parahippocampal gyrus are way more interested in spatial than non-spatial things, and in episodic than semantic things. And the combination of those two, spatial episodic, is what activates the hippocampus the most. So this is what the hippocampus is predominantly interested in. Turns out, for the cognoscenti in the audience, that there's a kind of a gradient along that the hippocampus is kind of like a sausage that stretches in humans out along a longitudinal axis. There's a very interesting story emerging about sort of what happens along that axis. It's so interesting that, 50 years after I did my PhD on it, I might actually have an idea what it actually means now. That was, my PhD project was to try to figure out that axis. It didn't figure out that axis at all. It just got me my PhD. But there's an interesting story emerging about the notion that the spatial part of the story is more in the posterior part of the hippocampus, and the memory part of the story is more in the anterior part of the hippocampus. This is an ongoing story, so to speak. I won't go into this last figure. I don't need to. It just makes the same point. All right, now what about remote memory? So the prediction of our approach was that if you could take somebody who's like really old, like me, and ask them to think about a memory from 35, 40 years ago that they hadn't thought about before, that you should still see hippocampal activation when they're thinking about these very old memories that they have retained. And this would not have been predicted by the standard model. By then, this memory should have been far away from the hippocampus. So we decided to test that. This is an already nearly 20-year-old experiment, obviously because there are so few subjects, and it actually still got published. We brought these people into the lab. We asked them, could you answer questions about, like, your first date or your first day in high school or whatever, something like that. And based on their responses, we then put them in the scanner and we gave them hints to retrieve those memories. And what did we find? What we found was both remote and recent memories equally activated the hippocampus. Now, there's a serious problem with this experiment, which I hope you picked up on, namely that we brought everybody into the lab the day before. Like, what were we thinking? Not clear. So we decided to do it in a different way. We created what we call the spousal control replication. In this experiment, what you do, and this is doable in Arizona, you go out and find married couples that have been together for like 40 or 50 years. You bring them into the lab. You separate them. You ask one member of the couple, do you think your partner could answer questions about blah, blah, blah? Okay. You use that to test the other one, who never got asked anything or was ever. All right, that's the spousal control. When you do it the right way, guess what? Looks exactly the same. So this basic result that the hippocampus is activated by remote memories as long as they're interesting enough and have been preserved in that way has been replicated by everybody. It's no longer a question of whether this happens. There's still some debate about why, but in our view, it's because the hippocampus remains involved every time a memory stays vivid, no matter how old it is. Here are some data from Morris Moscovich and Mary Pat McAndrews, basically just making that point, that what activates the hippocampus is the vividness of the memory. And again, no matter how old the memory is, if it's vivid enough, the hippocampus is still engaged because you need the hippocampus to sort of dredge up all those details that are still stored out there in cortical sites. Okay, this is the second summary. The hippocampus makes spatial, contextual, and episodic memories and is engaged throughout the life of a particular memory. Access to these details in this memory require the hippocampus, though knowledge about most of the details actually resides elsewhere in the brain. So people talk about the hippocampus being an index to the details that sort of make up our life. I'm not sure that's exactly the right language, but that's the way people are talking about it. And these indexes then contribute to the reactivation when one recalls a memory, so that it sort of pulls out all the different bits and pieces that are the memory. Okay. Now, given that background, can we change memories? What happens with the notion that memories can be changed? In other words, can reactivating a memory actually change it or lead to the possibility of change? So let's go back to the traditional story. There's an event, you encode it, you consolidate it, it's a fixed memory. That's in every textbook prior to the last 10 or 15 years. Trouble is, it's wrong. What we now know is that if you reactivate a so-called fixed memory that has been fully consolidated, that memory becomes unfixed in some way. And in the normal course of events, when you unfix a memory in this way, it gets reconsolidated and refixed and everything is fine. How do we know this is the case? Because if at this point, after you've reactivated a fixed memory and you unfix it, if you inject protein synthesis inhibitors, now mind you, this is the kind of experiment that you can only do in rats, although something similar is being done in humans with propranolol and other things like that. These protein synthesis inhibitors, acting on this unfixed memory, block reconsolidation and possibly delete the memory. Well, you can imagine how exciting this was to people who were interested in sort of erasing traumatic memories and so on, and there's a very active field trying to investigate those things. Our thoughts about this result, and I'm sure you all have the same thought, was, well, that's kind of a crazy way to build a memory system. If every time you retrieve a memory, it's in danger of being wiped out. I mean, there must be some kind of adaptive function to this way of doing things. So the notion of adaptiveness here is that memories are reactivated because you're probably, for some reason, brought back into that situation, or you're thinking about it again, or you're in a situation that reminds you of it, right? That's what reactivates the memory in the first place. Now, maybe in this situation, there's something new has happened. You want to update your memory with this new information. So I haven't been in San Francisco for a few years, probably, maybe a year and a half. There are a couple of new buildings here that weren't here before. They're now part of my map of San Francisco. I've updated that map because I've reactivated it. I'm back on site, and so on. So this updating and this sort of incorporating new information allows our memories to be living beasts, so to speak, that remain adaptive and functional even as the world changes around us. So you have a transformed memory as a function of this reactivation. Okay, how can you show that this is what's going on? So we designed this paradigm. This is a kind of a list learning experiment in some sense. Subjects are brought into the lab. There's that blue basket up there in the left-hand corner, I think you can see. It's full of real objects, you know, things like a comb or cell phone or something like that, or a flashlight and so on. And they're pulled out one by one, and the subjects are taught, you know, to remember these objects. And they get 4 trials, and they have to recall them. It's a standard list learning experiment. 2 days later, they're brought back. Well, they're given either 4 trials or to a criterion of 17 out of 20. 2 days later, they're brought in the standard experiment. They're brought back, and they're split into 2 groups. Well, 3 groups, but I'll leave the interference control out for now. One of them is the reminder group. They're brought back to the same context, the same experimental room. They have the same experimenter. We actually ask them, do you remember what you did here the other day? We don't ask them to recall anything. We just you know, jog their memory a little bit to reactivate it, we thought. The other group, the no reminder group, were brought to a different room with a different experimenter, no question, and both of them were then given a second set of objects to learn. In a different way, no blue basket, now they're spread out on a table, but essentially the same story. Our assumption was that for the reminder group, whose first list and all the objects on it had been reactivated, that when they are learning the second list under those conditions, some of those items from the second list are going to be incorrectly attributed to the first list. They're going to be added to that first list memory. This should not happen in the no reminder group that hadn't reactivated their first list memory. Obviously, again, that's the way it worked out, again. What you can see is that the reminder group, the red bar, shows you the percentage of items from the second list that were inappropriately attributed to the first list. That's 4 or 5 of the items. It's quite a lot, right? You don't see any such thing in the no reminder group. We unpacked it a little bit. We asked if you separate out the 3 cues to the reminder, the context, the experimenter, and the question. Turns out in this experiment, the spatial context is the key. That's what triggers this sort of process of memory updating, or at least reactivates the memory in a way that it triggers the process of memory updating. So we then wanted to ask, what's going on in the brain when this is happening? We redesigned the experiment. I should, by the way, give credit to, you know, as everyone has said, all the work is done by other people. These are 3 of my Kate Simon was the graduate student who really did most of the work, and Rebecca Gomez and Paige Scalf were faculty colleagues. Rebecca's still in the department. Paige has moved on elsewhere. We redesigned the experiment. We were now using sort of visual images of cards, of the objects, not the real object. And we trained them these objects in association with their sounds. So if the object was a saw, they also heard the sound of a saw. And if it was an alarm clock, they heard the sound of the alarm clock going out. And they learned this pairing. The objects were the key, but they were associated with sounds. After they had learned that, to one strength or another, again, 3 trials were given, or I think there were, well, whatever. Then 2 days later, they came back and we put them in the scanner, and we used the sounds to try to reactivate the memory, the sounds associated with those objects. We played back 10 of those sounds on the assumption that it would bring back to mind the objects in question. We then trained them on a second set, as we had done before, and then we came back 2 days later outside the scanner and asked them to retrieve the first set. And again, the question is, will they take some of those second set items and put them into the first set when we ask them to retrieve? And the answer was yes, but in an interesting way. Not every subject showed the effect. This was actually lucky for us in some way because it gave us the ability to make certain comparisons between subjects who showed the effect. Those are the ones on the left-hand side of the slide. Those are, again, the red bars represent these inappropriate attributions of set 2 items to set 1. So 6 of the subjects showed a lot of that. 5 of the subjects showed virtually none of it. So we have 2 groups, a group that shows updating and a group that doesn't. What's going on in the brain that distinguishes these 2? How can we tell what's going on? So we looked at what was going on in the brain while we were reactivating the first set of objects with the sounds associated with them. And what we saw was there was no relationship at all between how much activity there was in the auditory cortex and subsequent memory updating, subsequent intrusions. So it wasn't how much auditory activation there was. However, there was a negative correlation between visual cortex activity and intrusions. What does this mean? It means that the more visual processing, the more robust the visual processing of these sound sort of visual pairings during memory reactivation, the more accurate their memory was going to be in set 3. So the more robustly they reactivated the memory, the less likely it was to be intruded upon by this new set of objects that they were learning. Okay, that's one effect. Triggered by sounds, this effect presumably reflects the fact that this reactivated the visual memories attached to those sounds and so on. Strong reactivation made it harder for set2 items to intrude. However, as you saw, some of those set2 items did update set1 memory, right? At least for some of the participants. So we then could ask the question, what was going on in the brain while those subjects who showed this intrusion effect were encoding the very items that got intruded? What was the signature of this kind of memory updating in the brain? I'm cutting to just one result out of many. This result shows us that in the temporoparietal junction, the TPJ, there was a difference between activation to the items that were subsequently correctly attributed to list 2 or incorrectly attributed to list 1, the intrusion items. And the result was that the more TPJ activation we saw, the fewer intrusions we got, the less updating there was. How do we interpret this? You're all, I'm sure, wondering this very question yourself. We assume that this relationship between activity and the TPJ, which some people, I mean, the TPJ is meant to do many things, but amongst the things it is assumed to be doing is reflecting the extent of processing of details. How many details are we attending to? How many details are we remembering, and so on? How many details are we retrieving? So the likelihood, so there is this relationship between the processing of detail and the likelihood of a reactivated memory being remade or updated as a function of current experience. So here's a kind of a sketch of a story for what we think is going on. When you reactivate a memory, you can do so with sort of a range of detail. I mean, you can do it either with lots of detail, very crisply and clearly. That should make it easy to compare and differentiate from current experience. Or you can reactivate it with rather low detail, which ought to make it harder to compare and differentiate from current experience. This is going to lead to differences in how much prediction error there is, so to speak, a mismatch between current situation and what's been reactivated. And that amount of mismatch or prediction error drives the process. It determines whether memories are going to get put together or whether they're going to be kept apart, right? In this kind of classic dual thing that's going on in our brain all the time between stuff that is activated because we're thinking about the past and stuff that's happening right now in the present. One can also use terms like pattern completion and pattern separation to discuss this. Now I'm going to give you a kind of a different way to think about this intuition visually. So imagine you were invited to a destination wedding in some Caribbean island like this, right? And this was the scene that you saw. And you form a memory of it and so on. And then time passes and the memory, you know, fades into somewhere in the back of your head. Sometime later, you're lucky enough to get invited to another such wedding. I use the word lucky here advisedly. You're lucky enough to get invited to yet another wedding, and being there is going to reactivate, you know, your memory for the first event. Now, here's where it plays out. Either you can reactivate that first wedding you were at in exquisite detail. Pretty easy to see these are not the same thing. Or you can reactivate it in a kind of a hazy way, with not very good detail. Not so clear that that isn't, well, you can tell from the look of the chairs, but because I didn't pick a good example, but it's not so clear that this isn't the same event. So as a consequence, you can imagine that the new event could easily send some information over to enrich or kind of build up again the old event, but that's not going to happen in this case. They're going to butt heads and keep each other separate. So that's the intuition, that depending upon how much detail you reactivate a memory with, it's going to determine whether that memory is going to be tampered with, changed by current experience. That's the intuition. So old memories can apparently be remade by new experiences in some interesting way. Can we do this intentionally? Can we actually make this happen? Well, I'm going to show you one study. This is the last thing I'm going to tell you about, barring some closing comments, suggests that we might be able to do this. And we did this experiment during sleep. So here's what we did. It's been shown in a number of labs right now that during sleep, we spontaneously reactivate, you know, things. We replay them in our minds. I alluded to this earlier. There have been a number of experiments, and Ken Paller is one of the leaders in this, and Jan Born in Germany is another, where you can target certain memories for reactivation by training people those items in association with sounds or with smells, and then playing those sounds or putting those smells into the room while they're sleeping, and those will reactivate the memories without waking a person up, and then something can happen to the memory. That's already been demonstrated. And people have shown that when you do that, you can actually strengthen memories by reactivating them during sleep. We were asking a different question that might be more sort of clinically relevant. We were asking if you could cause people to forget memories during sleep. So here's what we did. First of all, we brought subjects into the lab, and then we taught them in a directed forgetting experiment, what we call a forget tone task. How many of you know what directed forgetting is? Okay. Basically, you give people a list of words, and some of the words are followed by a tone, and you tell them, forget about those words. You don't need to remember those. Just remember the ones that are not followed by the tone. Right? Then you come back later and you say, I was kidding. I really want you to recall all the words. And then what you see is that actually they don't recall some of the so-called forget ones as well. Right? So this tone came to sort of inhibit processing of those words, and it's called directed forgetting. We used that paradigm. We weren't interested in the words. We were interested in the tone. We wanted to create a signal that we could play back in sleep that basically said, forget. That was the purpose of the first part of the experiment. Then shortly afterwards, they learned about a bunch of objects, and they learned them in a kind of a 4-quadrant way that they learned not only the object, but which quadrant it was in. Right? Then we come back during sleep that same night, and we target during slow wave sleep, we play back 5 of the sounds associated with those objects. Again, we were using objects that had very typical sounds. On the assumption that playing back those sounds during sleep would reactivate those particular memories. We then followed that with the forget tone. Again, if the subject woke up, they're out of the experiment. So this was all happening while they were asleep. The hypothesis was those items that had been reactivated and followed by this forget signal would be less likely to be remembered when we came back to test them. And that was done 7 days later, and that's exactly what happened. If you look at the black bars on the left, the reactivated objects, there was a precipitous drop from 60% recall to 40% recall for the reactivated objects. And this effect was somewhat dependent on the strength of the learning of those objects. So the objects, as I said, were trained over a number of trials, so some objects were sort of learned better than others. And the stronger they were learned, that's the 75%, the somewhat weaker effect, but you still see the effect. Again, reactivated objects followed by a forget signal during sleep are less likely to be remembered. And as well, those objects that they forgot, so to speak, as a function of the signal, they also lost the spatial information. And that wasn't the case for the control objects, even if they weren't recalled. Because when we subsequently gave them on a recognition test, even if they hadn't recalled them, they still had some idea of where they were located once we gave them on a familiarity basis. So we think that this demonstrates that you can actually effectively change memories through this reactivation method. So what's next in this world? Well, was sleep important? We don't know. We did the experiment in sleep because it was easier to do. Even though it's not easy to do a sleep experiment, at least the subjects weren't doing other stuff while we were trying to train them these effects. Could we possibly activate 2 unrelated memories during sleep and cause them to become associated at that time? In other words, could we induce learning during sleep in a way that hasn't been done before? We don't know. Hasn't been done yet. Could we actually train the subjects with a forget signal and a remember signal quite separately and drive some items down and some items up simultaneously in the same experiment? That also remains to be done. We haven't done that. We haven't done most of it. Could we reactivate and then alter meaningful and possibly harmful memories from real life? That's the real target here. And that question, again, remains to be answered. All right, here are some take-home messages. I have no idea where I am with time. People aren't fainting yet? Okay. Memory engages multiple brain systems. These different systems have distinct properties. They enable different kinds of behavior, and they differ developmentally. The hippocampal memory system is concerned with space, context, episode memories, and is engaged throughout the life of any vivid memory of this type. Rich access to details requires the hippocampus, even though most of these details are actually represented out there in other parts of the brain. You still need this index or this access road to get to them. Reactivating such memories renders them fragile and open to being changed by current experience. And whether or not this happens is a function of the strength and detail of the reactivation. And we don't know the parameters yet that sort of force one or the other outcome. And this memory malleability opens up enormous clinical possibilities, as well as having implications for various forensic and legal issues. And changing memories could, it turns out, play an important role in therapeutic efficacy, and that's something that I'm kind of working on now. All right, I'm going to close with a couple of small remarks. This is a picture that I took in 1970 when I was a postdoc in Prague. This is an old man sitting on the bank of the river. How many of you have been in Prague? It's a beautiful river that runs through Prague. It's the Vltava in Czech and the Moldau in German. He was sitting there, and I lived actually in that neighborhood. It was a little bit outside the city, and I saw him there on a couple of occasions. And so finally, when I had enough energy, when I was confident enough in my Czech to ask him the question, "What are you thinking about?" And I'd been there a couple of years, so I was pretty good in Czech at that point. And here's his answer. "I'm thinking about my wife. We always used to come to this place together." I was too stupid at that point to realize he was telling me something very important about memory. That place is an indicator of memory. It's a tag, it's a signal for reactivating memories. And he basically told me that 2 years before I had similar thoughts myself. All right, I can't close this talk without commenting on Tolman, the father of cognitive maps, right across the bay here in Berkeley. I think those of you who don't know enough about Tolman, perhaps you should learn a little bit about what kind of a man he was. In addition to originating cognitive maps and having an enormous impact on the field, he also did the following, or the following was done to him, you might say. Interestingly enough, Tolman believed that people could form either broad or narrow maps. He wrote about this and the kind of the political implications of his own work. And he said that under some conditions, people form very broad maps, which we would call a cognitive map, a rich cognitive map. Under other conditions, they form very narrow maps, which you might sort of talk about as like just a route. It's nothing like a map. It doesn't have anything like the richness of a map. And he pointed out that these broad or narrow maps could correspond to what we mean when we talk about broad or narrow-mindedness in people, the willingness to take a broad view or a very narrow view on things. And he indicated that stressful conditions predisposed to narrow maps. What he didn't know, and what shows him to have been absolutely prescient in thinking these thoughts, was that too much stress damages the hippocampus. We know now that the hippocampus is loaded with stress receptors, and too much stress actually can damage that system. So indeed, stressful conditions make it less likely that we're going to form sort of a broad understanding of the world, and make it much more likely that we're going to form a narrow understanding of the world. That has very deep implications for lots of things that are going on in the world now. And I will close with this quote that you should read, and with thanking the organizers for inviting me to address to this Kavli audience. Thank you very much for that. Some of my collaborators and some of my funding sources, and thank you for your attention, and enjoy dinner.